I'm Dr. DeBusk, and in this video I'm going to talk about the internal structure, development, and specializations of leaves. We'll begin with the outside of the leaf, the epidermis. Thin, flat foliage leaves, optimal for light interception, have a large surface area which water can be lost through transpiration. It is a serious problem if the soil is so dry that roots cannot replace lost water. Leaf and stem epidermises are similar. In most leaves, the number of stomata is much greater in the lower surface than in the upper surface. This is to reduce water loss due to sunlight heating the upper leaf surface and prevent fungal spores from penetrating. For plants adapted to dry conditions, leaf stomata are frequently sunken into epidermal cavities that create a region of non-moving air, stomatal crypts, as in this oleander leaf. Water molecules that bounce out easily re-enter the stomata. The leaf epidermis is often hairy. The trichomes affect the leaf biology in numerous ways. They provide shade in the upper surface of the leaf. They prevent rapid air movement, slowing water loss from stomata. They make walking or chewing difficult for insects. Glandular trichomes secrete powerful stinging compounds that deter even large animals. Leaves of this lamb's ear is so densely covered with hairs that they often appear white. The hairs reduce the amount of heat these leaves absorb from sunlight. Plants of Sempervivum are native to high alpine regions where ultraviolet radiation is intense. The hairs of the leaf epidermis reflect part of the ultraviolet radiation away from the plant. The ground tissues interior to the leaf epidermis are collectively called mesophyll. Along the upper surface of the leaves is a layer of palisade parenchyma, or palisade mesophyll. It is the main photosynthetic tissue of most plants, so it will be on the part of the leaf that receives the most sunlight. Some plants have palisade mesophyll on the top and bottom. In the lower portion of the leaf is the spongy mesophyll. It is open, loose parenchyma that permits carbon dioxide to diffuse rapidly away from the stomata in all parts of the leaf surface. Vascular tissues are between palisade parenchyma and spongy mesophyll. A eudicot leaf usually has one large midrib or midvein with lateral veins that emerge and branch into narrow minor veins. The midrib and lateral veins contain xylem on the upper side and phloem on the lower side. A bundle sheath may be made up of fibers, may be arranged around the vascular tissues. Larger veins often have a mass of supportive fibers above, below, or both the bundle sheath extension. Petioles are part of the leaf. They are the transition between the stem and the lamina. The arrangement of tissues differ at the two ends. The epidermis often contains fewer stomata and trichomes, and the petiole mesophyll is rather like cortex. Parenchyma may be present if the lamina is heavy. 1, 3, 5, or more vascular bundles branch from the stem and diverge towards the petiole, called leaf traces. This section of stem has 11 bundles, 3 of which produce leaf traces that enter the petiole. Later, the leaf traces diverge and spread throughout the blade. These traces are drawn without any branching or merging, although both are possible. In many species, the petiole also bears 2 small flaps of tissue at its base called stipules. Leaves are produced only through the activity of a shoot apical meristem. At the base of the apical meristem, cells anterior to the protoderm grow outward, forming a leaf primordium that extends upward as a narrow cone. During this stage, the primordium consists of leaf protoderm and leaf ground meristem. A strand of cells in the center differentiates into provascular tissue and then into primary xylem and phloem. This forms a connection with the young bundles in the stem. As the leaf primordium grows upward, it increases in thickness, establishing the bulk of the midrib. The young leaf consists of a midrib and 2 small thin wings of lamina tissue. All cells in the wings are meristematic. Their division and expansion enlarge the lamina rapidly. During lamina expansion, the stomata, trichomes, and vascular bundles differentiate. The petiole becomes distinct from the midrib. When complete, the leaf is still very small, much less than 1/10 its mature size. Full expansion, typically in spring, occurs as water enters and expands the cell. In many perennial plants, leaves initiate in the summer or autumn. They become dormant as part of a resting terminal or axillary bud until the growing season begins. When the bud opens, young leaves rapidly expand and mature. Aphids and other sucking insects typically attack young leaves and stems that have not yet developed any protective sclerenchyma around the vascular bundles. Monocot leaves, like those of eudicots, are initiated by the expansion of some shoot apical meristem cells to form a leaf primordium. Apical meristem cells adjacent to the leaf primordium grow upward along with it, becoming part of the primordium. It gives it a hood-like shape. The primordium becomes tubular, forms the sheathing leaf base, while the upper portion forms the lamina. Those with linear strap-like leaves grow continuously from a basal meristem. It remains active so that if the lamina is removed, more can grow. Tissues differentiate just above the basal meristem. Now I'm going to talk about some of the common specialized leaves. Numerous adaptations permit plants to survive in desert habitats, one of the most common being production of succulent leaves. Succulent leaves are thick and fleshy. They survive in desert habitats. They have reduced surface to volume ratios, favoring water conservation. The mesophyll contains very few air spaces. The mesophyll is more transparent, so photosynthesis may occur deeper in the leaf. Leaves of Senecio rotundifolia are spherical, giving them an optimal surface to volume ratio for conserving water. Leaves of the succulent Denturanthus is hemispherical and attached to the opposite leaf, greatly reducing the exposed leaf surface. Interior tissues are mostly water storage parenchyma. Their low levels of chlorophyll result in low levels of photosynthesis, but a lack of water is more significant danger for these plants. Each plant consists basically of 2 leaves and a microscopic stem. The root systems, which is not visible, is substantial. This agave has survived a wildfire in West Texas because its succulent leaves protected the stem from the heat of the fire. The center of the plant, especially the shoot apical meristem, is still healthy. Containing so much water, the leaves heat up slowly, and before the innermost leaves became hot, wind had blown the fire farther on and the stem and apical meristem were saved. The leaves even kept the ground at the base of the plant cool, protecting the roots. Sclerophyllous foliage leaves have sclerenchyma as a layer below the epidermis and in the bundle sheets. In some species, they are perennial. Their hardness makes them highly protected. Leaves of conifers are sclerophylls with thick cuticles. Their epidermal and hypodermal cells have thick walls. They usually contain unpalatable chemicals. The shapes can be needle-like or flat and scale-like. Yucca leaves are excellent examples of sclerophylls. They are tough and fibrous, and the cuticle is thick. These features reduce the light reaching the chlorenchyma, but the plant growth is probably limited by the scarcity of water rather than the reduced photosynthesis. These leaves may function for 5 to 10 years or more. Bundles of fiber occur just interior to the epidermis, and the thick mesophyll surrounding many vascular bundles. Stomata occur in long, shallow grooves that run between the fiber bundles. Even the vascular bundles of these sclerophylls have fiber sheaths. Bud scales are small modified leaves that form a tight layer around the stem tip. They protect dormant buds in the winter. They have short or absent petioles and are often waxy and tough. Some are hairy and may have thin, corky bark layer. Spines are modified leaves of axillary buds. They have needle-sharp projections. They are protective and made primarily of fibers. The lignified walls make them hard and resistant to decay. Spines of this barrel cactus are modified leaves, and the cluster is an entire axillary bud. The tendrils of many species, peas, cucumbers, and squash, are another form of a modified leaf. Unlike photosynthetic leaves, tendrils grow indefinitely and contain cells that are capable of sensing contact with an object. When the tendril touches something, the side facing the object stops growing, but the other side continues to elongate, causing the tendril to coil around the object and use it for support. A distinct type of leaf anatomy occurs in plants that have a special metabolism called C4 photosynthesis. These leaves lack palisade parenchyma and spongy mesophyll, having prominent bundle sheaths composed of large chlorophorous cells. Surrounding each sheath is a ring of mesophyll cells. Kranz Anatomy adapts C4 plants to arid environments. Plants growing in habitats poor in nitrates and ammonia evolved insect traps. The captured insects are digested as a source of nitrogen. The leaf appears highly modified, but is actually similar to many foliage leaves, except the lamina is tubular and secretes a watery digestive fluid. The epidermis is absorptive. Trap leaves can be classified as either active leaves that move during capture or passive leaves incapable of movement. That includes this section on the internal anatomy of leaves and the types of specialized leaves.